Okay, go for it. Okay, be ready with the code. Okay, let's start. Okay, very good. I was not perfect, so
Professor,
mhm.
I think my computer has some problems. Can I exit this Zoom and
At least
connect?
at least I can see you. Currently you are connected to Zoom, but you can try to connect to the Kahoot.
I connected, but there 렉이 좀 생기고 있는 것 같습니다.
Zoom에서.
네.
Okay, try again to go out and come back. But can you see the Kahoot? Can you continue to play Kahoot?
예,
Kahoot, do you use a computer or your cell phone?
I use my computer now.
Then try again. Connect. Go out and then come back quickly.
Okay.
Okay. So while we are waiting for the Yonghun, so stem cell is a pluripotent, and unipotent means it can make only one type of cells. So muscle cell, if they divide, they can make only muscle cell, and the neuron cell, they can make only neuron cell. That's a unipotent. Multipotent means it can make more than one, but not every the cell type in our cell. Okay, pluripotent is much bigger than multipotent. Multipotent usually one cell can make 2, 3, 4 cells. Let's just say usually we call blast. Blast means it's a kind of the seed or the starting precursor cells. And those cells can make a different 3, 4 different cell type, but it cannot make hundreds of different cells. Our body may have more than 100, maybe up to thousands of different cell types, and only stem cell can make a pluripotent, and it can make every type of cells. Then totipotent here. Anybody that knows the meaning of a totipotent? Nobody knows? Okay, totipotent means it can make every cell type in our body, but at the same time, it can make the whole body. Full life form it can make. That's a totipotent. I will explain that later again. Anyway, stem cell is a pluripotent. It can make every part of organ, every type of cells in our body. That's a pluripotent. Young is back, so we are moving to next problem. Okay, IPS is an induced pluripotent stem cell. Induced means it's kind of artificial, the stem cell. But if it is a perfectly well made IPS, then it should be same as a stem cell. Okay? IPS is also pluripotent. Today I'm not using the earphones, so I guess you can listen to the sound of a Kahoot too, correct?
That's correct.
Okay.
Yes.
It's more fun with the sound. Okay, 100% correct answer, but two of you could not answer. Do you have some of you have a problem to answer the Kahoot for now?
I clicked C, but I think there is some delay for internet, so it mischecked the answer.
Strange. Is that my problem or your side problem?
No, that is my problem.
Okay, and try to answer as soon as possible. Okay, so
Okay.
DNA methylation happens on C, cytosine. Not every cytosine, but methylation happens depending on sequences. Okay, not every C, cytosine connected to G, C, G, then this C can be methylated. If this is C, G, C, then this C does not get methylation. Only this C, C, G, C, CG position of C can be methylated. Okay, and if I write this way, then two-way is the different directions. This direction, if I write, then this side is 3' or 5'.
5'.
Second,
5'.
this is supposed to be 5', and starting from the 5', we are moving to the 3'. Okay, always we write this direction. So CG is the position of methylated, and this is a little difficult to pronounce. CG, maybe it may sound like computer graphics. So usually we put E inside the in between here. So CPG, that is the usually the site we call CPG site on C. That's the methylation site. Okay, very good. Gene expression means this is a transcription. Okay, gene expression means the transcription down. DNA replication is a DNA copy. DNA copy does not have an effect on methylation. Protein synthesis is done by ribosome. So DNA methylation is affecting gene transcription, okay, or the gene transcription down. That's why we call gene suppression. Okay, the answer for this histone methylation is happening. The result, we don't know exactly what is going to happen. So I tried to make a multiple choice, but now the answer can be either transcription high or transcription low. We don't know. But the methylation itself on the histone, it's difficult to predict. Okay? If I choose this multiple choice, I should pay for this Kahoot. So I didn't choose that. But anyway, answer for this question is histone methylation, we don't know the result. It can be either going up or going down. Okay? Methylation on DNA, yes. It's almost, it is gene suppression. But histone methylation, it can be random. And also it can be affected by other factors together. Okay, acetylation is different. If the histone is acetylated, then transcription becomes higher. Okay, this direction is clear, but as I said, methylation, the effect is not clear. Okay. Okay, here the answer is two. One is usually the methylation pattern. Methylation is low, and telomere is long. Okay, these two is the right answer. And when embryonic stem cells start to differentiate, then they divide 2 cells and 4 cells. Then DNA methylation slowly goes up, and also telomere, the size is also becoming shorter and shorter. Okay. Okay, cancer cell is very similar to embryonic stem cell. Okay, that's why if you wanted to be young and then attack those telomere or methylation, then there is a very good chance to become cancer cells. Answer is the same as DNA methylation in general, that's low. And also telomere size is getting longer too. Okay. Any questions so far for this quiz? This is just to check whether you studied those epigenetic material reasonably or not. Don't be shy. If you have any question, then you can give me a question now. Okay, for the midterm exam, before I forget, I recommend you to read the book, the 유전자는 알고 있다. That book is a very good reference book for this epigenetics. So I may choose 1 or 2 problems from that book too, general one. So I recommend you read that book. That is a very good book to read. Okay. So I think this is the last problem for this. Oh, one more. Okay, sorry. It can be either transcription high or transcription low. The answer is half and half. Half of you succeeded, half of you failed. HDAC is a histone deacetylase. Okay. HDAC is high, then the acetyl group is removed. Then the transcription is down. HDAC inhibitor is added. Then deacetylation is blocked. Then acetylated histone amount is going high. Okay. Then histone is highly acetylated. Then transcription is high. So this is negative of negative. That's why it's a little confusing. But this is important. And this HDAC is popularly being used as an anti-cancer drug. And this is the important part you should know. Okay? If it is not clear, then this part you should review again for the study. Okay. Done for today's case. And let's move on. Okay, now we go back to today's class. Can you see the screen?
Yes.
Yes.
Then let's move this way. Okay, today's topic is epigenetics. And in epigenetics, let's say, the egg cell meet sperm cells. There are maybe 20, 30 sperm cells around one egg cells. Only one lucky guy, girl, I don't know, one sperm can enter the egg, and then it can start the new life. And by the way, this image here, what do you think? This image is an optical image, microscope image? What kind of a technique generated this image, do you think? Fluorescence image or bright field image, optical image? This is a electron microscope image, EM image. Okay, so the size of this head group may be around 10 micrometer, several micrometer, and we cannot see these small details on the cell. So this is electron microscopy image and artificially colored. This is not optical image. In optical image, we cannot get its high resolution. Looks like a quite dynamic, but this sample should be highly dehydrated. The water was removed, and also maybe the metal was coated on the surface. So this is kind of a ghost image, not live Earth image. Anyway, one fertilized egg cells start, then the blastocyst is formed, divided into 4, 6, 4, 8, 16, and then divided hundreds of cells, and then they started to make this kind of form. And there is inner cell mass, and also some other cells outside here, so-called the trophoblast cells. To make a full baby, we need both this inner cell mass. This inner cell mass make embryonic stem cell. Okay, these cells can make every organ in our body, every organ or every cell types. That's why we call this embryonic stem cell is a pluripotent, but it is not totipotent. Without this trophoblast, it cannot make full individual because this trophoblast make this part, so-called placenta, in Korean, 태반. When the embryo grows, this part of the cord, umbilical cord, so-called the petul, at the end there's a placenta, and this placenta, the embryo put this placenta onto mother's organ, and then suck up a lot of the nutrient. Okay, so embryo does not fully believe mother, and this is kind of a competition between baby and mother, how to manage the nutrient in the mother's body. Okay, so that's why this is kind of a lunchbox for embryo, and the embryo does not believe mother's the offering, so it attached that the placenta to mother's body and suck up a lot of those energy from mother. Okay, so that's why usually during those pregnant period, the mother usually becomes diabetics because this embryo releases a lot of factors to let mother to release a lot of glucose into the blood. That's why the mother suffers from high glucose the symptom, that is diabetics. Okay, so that's why without this trophoblast, placenta cannot be made. Then the baby cannot survive. That's why embryonic stem cell itself can make the body of an embryo, but it does not make this placenta. That's why it cannot be, it is not totipotent, it is a pluripotent. Okay. Now you learned those concepts of totipotent and pluripotent, then you should be able to discriminate. Now for this week, we studied 6 weeks together, and I tried to change a little of this course by giving you a little less burden. In a sense, I tried to define those questions and then wait for your answers. And I felt your answer was a little passive, and also you gave only one answer for each questions. So this week I tried to give you open questions. Whether I give you a question, you generate questions. Again, the result, I was not fully satisfied. Most of you just gave one question each for one problem. My expectation was a little more creative and thoughtful questions. Giving good question is very important because that reflects that how much you studied and also how much you thought about the topics. Yeah, some of the questions was quite good, and we are going to discuss with them. But some questions were just randomly tested, okay, without too much thinking. I want to know just something, then that does not, it's difficult to figure out how much you understood by yourself. Also, I expected you give the answers to your friends' questions. Then it's an active discussion is ongoing. That was something I expected. So this week, maybe the practice was not fully done. So I did not usually, when I see your questions and I give a grade for each of your questions. So this week, I do not discriminate much. Okay. So this week, from this week, we'll start to practice active class participation. So let's just say this is one of the question was given by Young-hun. If you don't want to give your name, then I can hide this, but nothing much to hide here. So question here, then you started to answer first. Okay, either verbally or you open up the chatting window, and then you give your answer by typing your answer by in chatting the window. And our TA, the Xiao, will count your answers, and I will also grade whether you are actively participating in the class or not. Okay, so usually in the last class, last year or 2 years ago, there were class size was much bigger, and there was a very active the seniors. They gave almost too much answers and too active, and that overwhelmed those juniors a little. But this year's class was relatively a little not super active, so I wanted to boost a little of this activity. So from now on, so by looking at each questions, you guys will start to answer quickly. Okay, we start with this. Young's question. I will not read. You read and then you start to answer. Either verbally or you can type your answer in chatting window. There are more than 20 questions today. You should hurry. Okay, some other mechanism was given by the Dong. So let's say microRNA or siRNA is something we did not learn so far. Yeah, some other method is also available. But now here the question is whether DNA methylation is it the only way, and that's maybe the Dong's answer is the related to this. But how about adenine or guanine? Okay, this question, let me try to help you out. So maybe we need to use this. Okay, this is basic structure of DNA and RNA. Now here, this is a DNA structure, APCG, and this is a RNA structure. Okay? And only difference between DNA and RNA is that T and U. U does not have this methyl group here. T has this methyl group here. Now, two fused ring, this structure A and G, we call purine. Okay, this is adenine and this is guanine. This is purine. And one 6-membered ring, this is a pyrimidine. And now one more methylation can happen to this C, and the site is very similar to E. Okay. So this site, this is methylated. Watson and Crick base pairing happens in this side. So that's why even though methylation happens here or here, at least the Watson and Crick interaction is not effective. That means DNA double-strand forming is not effective. That's why methylated or not, DNA replication can happen. Okay, open up and then just synthesize those the DNA replication or DNA recognition is not a problem. You can go ahead. Maybe by looking at different structures, adenine or guanine, or maybe even T is already methylated, so it can be a little difficult to do the methylation here, but other modification is always possible. Or even this methylated version can be changed into a hydroxyl group, aldehyde, acid, kind of a different modification is also possible. So there are currently 20, 30, 40 different modifications on DNA or even on RNA. Is every day new modification is being discovered. But so far, seems like this methylation on C is most abundant and very apparent. And the end result is also very clear. More methylation on DNA. That means gene downregulations. Okay? Gene downregulation means not DNA replication. Gene downregulation means RNA synthesis, transcription. Okay? Transcription is suppressed. That is the most abundant effect. That's why methylation, everybody that talk about it. Okay, let's look at next question. How about this question? There are many Cs, but the site should be random. It cannot be random, right? There should be some good control. Which C should be controlled, which should be methylated, and which should not be methylated. And any idea for this question? Only small methyl group. Then how does it stop transcription? And I said this methylation does not affect the Watson and Crick recognition. Then how does this small methylation can stop the transcription? Any idea? Okay, if I keep answering myself, then I should get the highest grade. Then total grade would be constant number. Then your grade cannot be so high. Okay, Juan said something. Select Shapasho. What is a Shapasho?
June automatically transmit English to Korean.
What
I
is the
was
original?
wondering to say by controlling selectivity of enzymes.
Selectivity. Okay.
Yes.
Selectivity by active the site. Okay, makes sense. Methyl group can block RNA polymerase binding, possibly. Steric stress, okay, maybe similar meaning of recognition or binding. So if you look at the gene expression, let's just say this is a DNA, okay, and usually there is some of the protein DNA binding domain bind here, and actually it should open up this DNA because the double-strand DNA is difficult to be read. Okay? So like a helicase open from the end position. But for making RNA, you cannot open the whole DNA. Maybe some middle part can open up this structure. And then other material, like let's just say activating domain and maybe the RNA, the polymerase, come and bind and then start this gene expression. Then if this is a 5 prime, one of them, let's just say maybe this the blue line is a 5 prime and this is 3 prime. And reading this 5 prime, no, opposite way, right? Template should be 3 prime. 3 prime DNA here, and then binding and activating, and then starting here. Then there should be some start position here. And template is a red one. Then RNA synthesis should start here as a 5 prime to move on down. Okay? So maybe there may be some the methylation happens on DNA, maybe binding over this binding domain or maybe activating domain, or maybe RNA polymerase is affected. Its real situation is much more complicated. So there is a binding domain, RNA polymerase, and a lot of activators, not single activators, and many small other, the small modules are binding to make this a big complex, not only linear DNA, affecting even higher upper stream of these DNAs. Some of those parts, promoter or enhancer part or silencer, these guys are competing to each other. And then when this three-dimensional structure is formed, then maybe RNA polymerase start to make RNA, okay? So this kind of very complicated complex is required. That's why if methylation happens, some of these positions, either enhancer or silencer, or maybe this binding site, each of them, they can change those binding affinity to the activator or different component. Okay, so this means this is not very simple, the mechanism, but definitely those methylations, some part here, they can change their binding affinity or binding selectivity. Okay? And that's why maybe the binding is weakened here, and they cannot bind here. This activator cannot stay here, and transcription cannot happen. Okay? This is a balance of many partners working together, and everything is activated. And it may not be 100% on and off, 100 or 0. It may be with some methylation, it becomes 20. Okay, more methylation, it becomes almost 1 or 0. Methylation decrease, then they increase maybe 60, 70, kind of quantitative changes. Okay. Now, methylation site-wise, in the quiz, I tried to tell you, if this is a 5', now it starts to go C and G. To make it easy to read, you can add the P here, so CPG, we call. Okay. If it goes this way, CPG, and even though there is another C here, it cannot be read because the next position is not G. Okay. If this is changed, BPC, it doesn't work. Starting from 5', we read to this direction, only CPG works. Okay. If this works, then how about the other side? The other side, this partner complementary with C, and C is complementary to G. The other side, now direction is changing. If this is a CPG, you don't read this as GPC. You read from this direction. So this is also CPG. Okay. So this kind of the symmetric structure we call palindromic. If it is a palindromic, that means once one CPG side is methylated, the opposite side also can be methylated. Okay. So that means one side methylated, the other enzyme is scanned. If this is methylated, it's methylated here too. Understood? Then, if you count this kind of the CPG site out of the total genome, what percent of CPG there is random? Come quick or type quick. If you cannot calculate, then you can give 1/4, 1/5, 25%. That means 1/4. If there is only C, if I X, that's a 25%. But that should be CPG, CG. It's not monomer. 40%? How you can count the 40% to the 1/12? Why 1/12? This is a quaternary. Yes, someone's answer is correct. 1/16. Whole combination is a 16, isn't it? It's a quaternary system. If it's a dimer, then whole combination is a 16. Then CG is a 1/16. Then 1/16. That's the right answer, isn't it? Right? Then that's about the 6%, 6 to 7%. Now, as I said, it's not GC. This is CG. That means that this is not combination. This is what we call. This is not Johab. This is a Sunyal, right? So that's why this is a 1/16. So if this is only 6%, then this should be random or not? That's an interesting question. And this is not random. So in most of those genes, okay, this CG sequence is quite rare. It's much less than 6%. But some area is highly condensed. Which area? This is the starting point of transcription, ATG. ATG is the code for methionine, and also this is starting point of RNA synthesis. That's why usually the protein synthesis, first amino acid is methionine, because this is starting point. This area, so this is upstream and this is the downstream. That means this area is binding site of those activator and also many different proteins for RNA synthesis, right? That's why this area is regulating of gene expression. That's why it is a highly condensed, highly enriched. The CG, CPG are the part. So this area we call CPG island. This definitely, this is much more than 6%. This may be 20%, 30%, highly enriched CPG positions. Okay. So this kind of the unbalance and this kind of a condensed area is important for gene regulations. So that means this is not randomly distributed. This is evolved to have high contents of a CPG on the own promoter area. So this area and down, this is mRNA sequence, right? That's why this part is a promoter area, but to decide whether to make RNA or not. So CPG island is highly enriched in this area. And also considering those complex structure, that recognition of methylation site is not random. So there should be enzyme which methylate these positions, DNA methylase. They recognize this structure, not only sequence, the three-dimensional structure, and then they methylate this area. Okay, next question. I wish some of you can answer this. Anybody? Sejin, you try. Sejin 선배, could you try? Sejin 선배, do you hear me? Sejin 선배 doesn't answer me. Oh, would you try?
I don't exactly know the answer.
I do not expect the exact answer. Just you try.
Can I say it by speaking?
Somehow your voice is breaking up. Can I hear the full sentence?
Oh,
Okay, Joan's opinion is maybe DNA replication, DNA methylation may be long-term effect. Histone methylation may be more reversible, maybe. And Taekjun says histone modification may be easier, maybe. So we just found, maybe the question is why this epigenetic modification should happen occurs in DNA and histone. Well, if it is designed by one designer, like a God, then maybe we can ask him. But most likely, this was trial and error process. It was established this way. So maybe the more reasonable the approach is maybe the end result and how this system is helping to control the current systems, or maybe how this system is we interpret the meaning of this rather than why. I think the answer may be easier to how this was formed and how this is affecting the whole systems. Okay, Young-hoon answered something, and it's not easy to follow all the you are writing here. Okay, Sejin also has some problem with the other machines. Okay, let's say there is, at least we found some of the DNA and also histone, the both of them is changing. The exact reason, we don't know, but both of them has some effect. And I summarized DNA methylation is gene activation down, and histone acetylation, gene expression up. Others, it's a mixed effect, we see. Well, some opinion of this, the different methylation pattern is more expensive. Why? This why is something we can answer. Would there be any procedure that could reduce the cost? Well, whether the one system is expensive or not, that's always relative number, right? This is more expensive than maybe the sequencing. Then last week we studied about the sequencing of the genes. Even with the next generation sequencing, our ability of sequencing increased a lot. And also very high number of sequencing is ongoing. Then per base pair price, this becomes very, very low. This is kind of the economic effect of mass production or mass testing kind of thing. Right? So that's a little difficult to directly compare, but at least comparing to sequencing itself, this methylation pattern cannot be just only sequencing. Now here is this cytosine, and if there is any methylation happens here, now methylation happen and methylation with methylation and without methylation, we need to distinguish them, right? As I said, this methylation on this position, it does not change anything for Watson and Crick, the effect. Then, if you do just the sequencing, you will get same sequence as C. So last week we learned about the Maxam-Gilbert method. The smart way of their method is by using some chemical treatment, they could selectively cleave some site of the base. That's how you can get those different signals. Here, similar things. If you use this chemical, bisulfite, only cytosine without the methyl group here, they can change this amine group with the oxygen. Amine was removed, and then they changed their identity from cytosine to uracil. And if this methylation is here, then this reaction does not happen. Okay, so you need to treat chemical treatment first by changing this cytosine to uracil, and then you read one is if it is methylated, then it is not changed. Then end result wise, you will get same sequence here. But if that site is not methylated, the site is changed. Originally C, they becomes U, then the sequence, the reading is different. Okay, using this comparison, you need to do two different sequencing. One is just sequencing itself. Okay, by dissociating this DNA, and then you amplify those sequences by PCR, and then you read those sequences. And compare that sequence with this bisulfite treated sample, then compare those two, then you can figure out which position of C is changed and which position is not changed. Okay, at least that requires double amount of sequencing, and at the same time that requires chemical treatment with this. That's why it is more expensive. And the need of handling this kind of sequencing is number-wise, maybe it's not very high number comparing to mass sequencing. That's why this method is much lower sample, but similar numbers of steps. That's why this method is much more expensive. Okay, so there are many CPG at the site is possible. Then usually, if this is a CPG, okay, then methylated, then no change. It's a CG. You will read. If there is an unmethylated, it will change into U. U is the same with the T in DNA. That's why if there are many sites, okay, this one hole represents one CPG site. Now one gene, and maybe starting point is here, APG site here, and select out those CPG sites and make this array, and then count whether which position is methylated, which position is not methylated. So this is a common way to show whether among these maybe hundreds of different CPG sites, how many percent of them are methylated. Okay, maybe this is the same site and different sample. Different sample means maybe different person or different tissue. Then they collect this site and then showing that. And this site's methylation is higher than this sample. That means sample B may have more gene expression here. Sample A has maybe lower gene expression. Okay, this is usual way of showing CpG methylation pattern. Okay, easily we can see here methylation is not 100%. Maybe this is a 20%, this is maybe 10%. And how much of those change is really affecting how much of a gene expression, that should be determined by experiment. Okay, any question for this, the images and also the method of methylation, the patterning? If not, we are moving on. Well, how this DNA methylation pattern is depend on external environment. In genetics, when we get the genetics or heredity means we get those genetic information from our parents. Okay, in that case, what we get is the sequence of DNAs. Sequence of DNA is very difficult to change. If the sequence is changed, we call mutation happen. Mutation is not easily happen. They need very serious chemical or photo reaction, and then the sequence can be changed. But this methylation pattern or histone modification pattern, they are relatively easier, and also they are reversible. Those methylation can be on, it can be removed. Maybe simple other example, like alcohol. So if you drink a lot, then DNA methylation pattern changes. How can it done? How can it be done? If you drink a lot of alcohol, if you think about the cell membrane usually hydrophobic material can penetrate. Hydrophilic material cannot penetrate, right? But ethanol is relatively very small, a little bigger than water, and partially hydrophobic, partially hydrophilic. So ethanol size, it can penetrate easily to membrane structure and loosen the membrane. So if you drink, I think every of you has experience of drinking the alcohol. If you drink, we feel loosened, right? Not tight. So physically it is happening. All our membrane structure are kind of loosened. Then penetration is increased, and even access to the DNA can be increased too. And maybe those binding affinity of DNA methyl, DNA methylase to those binding sites can be also changing. Those kind of physical environment, exactly how, we don't know. And also maybe it can affect those complex structure of transcription factors. They can be also either positively or negatively changing. Then DNA methylation, somehow it can be affected. Which direction, we don't know exactly. If it is really critical and bad, then maybe that kind of the changes, it should be fixed very quickly by evolution. If not, the individual cannot survive, right? If one person is keep drinking, then we get resistance to the drinking. We can be better and better for drinking. Means some of those environment, some of those machinery, they changes their sequences and structure and adjust the body, right? So if we expand this alcohol effect to different external environment, there can be many external environment: drinking, smoking, and eating what kind of food, or life pattern, like whether we do exercise or not. That kind of things, all of them, somehow they affect. And that the effect is residing in our body as a DNA methylation pattern or histone modification pattern. Exactly how, for which position, we don't know, or we know for long-term effect, there is some effect left over, and whether they are good or bad, we need to analyze it later. Okay. Now HDAC came out again. What is HDAC? If you don't know the HDAC, then there's no point to discuss. What is HDAC?
histone deacetylase.
Histone deacetylase. This enzyme removes acetyl from histone. Histone, I think I have some picture somewhere. Variety of so HDAC shows knockout means you remove HDAC from gene. Okay, knockout. Well, in the biology is usually handling the very big object. That's why sometimes the term we use is limited. So once we make one term, then maybe comparing to that, we make in and out kind of a different expanded the terminology we make. If you put in the gene, we call knock in. Okay, human body does not have any prophylized gene. If you put the prophylized gene into the human, then it's kind of a foreign gene, but DNA structure is the same. So we get new sequence in our body. We call it knock-in. We had HDAC in our body. If that gene is specifically removed from the gene, from the DNA, that is called as a knockout. Okay. Previously, the one of you said about siRNA or microRNA, different kind of a control. If you control those gene expression, means the RNA synthesis is increased or decreased, then we call, if it's decreased, we call knockdown. Then if it's a knockdown, if there's any method to increase those gene expression, how will you call?
Knock up.
Knock up maybe, right? Knock up is not a very popular word to use, but anyway, it is the way how we construct the biological terminologies. Once many people start to use that term, then it becomes, accepted the terminology. So there are many cases like that. So here is one case. Knock out means take out the gene. If you take out the HDAC, there's something happens in the cardiac symptom. Then acetylation and cardiac tissue is closely related. Well, this kind of argument is very difficult to make because, once you affect, our body is composed of thousands of different other machinery. This experiment is you just remove one machine out of a total system and see what happens in our body, right? So we believe our body is super complicated. That's why we may need to have 100,000 of different other protein, but surprisingly, we have only 20,000 or 30,000. So 20, 30,000 genes should do everything. That means one protein may do many functions. And different area, they have different functions. It's very possible. So that's why, whether by removing this HDAC, we got cardiac problem. Then acetylation is cardiac related the function. It's too general to say so. Acetylation can happen everywhere. Acetylation is relatively simple changes. Or methylation, it can happen everywhere. Just as an end result, it shows more effect in heart. Okay. That's all we can say. Anybody can answer for this? Okay, in the piece I said, cancer cell is similar to embryonic stem cell. Okay, embryonic stem cell, it should make every cell type in our body. That's why usually the embryonic stem cell, they need to express almost every genes in our body. Because it is kind of a master cell, we have limited numbers of genes, like 20,000 or 30,000. If it is a master cell, then the master cell should express almost everything, right? And embryonic stem cells function is, it's not rather than it has a specific skill, it's just proliferate and make more numbers and just prepare for differentiation. and it's very similar to cancer cell. So generally speaking, cancer cell also has low methylation, means many genes are activated. And also the telomere size is getting longer, and they can live forever. Okay. Now, for these specific cases, not every genes are increased, and some genes are actually decreased too. So if it becomes cancers, there may be two classes of genes we can think. One is suppressing cancer, one is increasing cancer. Then, if some genes are shut down, decreased, then those genes, we can expect those genes should be cancer suppressing genes, right? One typical example is P53. P53 protein is representative famous cancer suppressor gene. If P53 is active, then cells move to the cancer direction, then they die out by apoptosis. Then to become cancer, this P53 is kind of a bottleneck. Then this gene is shut down. Okay. HDAC inhibitor, if it can be used, HDAC inhibitor can be used cancer drug. That means HDAC inhibitors target should be HDAC inhibitors the function we saw is increasing gene expression. In cancers, those genes are tumor suppressing genes, and those are suppressed. That's why they became cancer. If you add HDAC inhibitor, those genes can be turned on by keeping those acetylation pattern more than other genes. That's why the cancer cells has good chance to be recovered or maybe self-dying, suicide. Cancer cells suicide is good for whole body. Anyway, we need to reduce cancer cells. Okay, so either they change their property from cancer to normal or they die. Okay, so this is what is going on. So in spite of some of those inactivation of HDAC, okay, now some genes still, they can be up or down, and then HDAC inhibitor can change those structure from one state to the other. Heterochromatin we discussed later. Okay, euchromatin and heterochromatin, and similar questions. Now, let's read those terms. In biological terms or the medical terms, there are many Latin or Greek, the suffix and also the prefix. So we had this term before. When did you hear this eu?
Eukaryotic.
Eukaryotic and prokaryotic. Then what is the meaning of eu? Prokaryotic means karyot means it's a nucleus. Okay, prokaryote means before nucleus. Before we have real nucleus. Okay, so u means usually u means this is real. The eukaryotic means a real nucleus containing cell is a eukaryotic cells. Okay, gene is a real, right? Now euchromatin means it's a real chromatin. Heterochromatin means this is something changed, different chromatin. That's a hetero, right? Same is a homo, hetero is a different, right? Heterochromatin means something changed from real chromatin. Okay. Then real chromatin is kind of open structure, and heterochromatin is something wrong, and they became aggregated. And if you look at electron microscopy images, this white structure is a euchromatin. It's kind of regular, real, normal chromatin. And this black side is aggregated chromatin. Something looks like sick or something wrong chromatin. Okay, these are heterochromatin. Now, HDAC inhibitors, if they are down, and if they are up, HDAC inhibitors up means acetyl group is removed, right? Acetyl group is removed, then histones charges become more positive. Histone is originally positively charged protein, and they make complex with the DNA. DNA is negatively charged, right? Acetylation is removed, then their charge plus minus charge is stronger, and they can be more condensed. More condensed gene is similar to heterochromatin. Once the gene is a heterochromatin structure, do you think RNA synthesis from this gene is easier or more difficult?
More difficult.
It's more difficult, right? If it's open up like this by more acetylation on histone, it becomes more open structure. More open structure, then access to make more RNA is easier. That's why if you look at this structure, euchromatin area is more actively RNA synthesis is ongoing. Heterochromatin area is kind of condensed. That's why they are more difficult to make gene expressions. Okay. Now, if I give you a question that here is embryonic stem cell, here is some differentiated cells, and draw those structure of euchromatin versus heterochromatin, which is wider?
Euchromatin.
Euchromatin is a white one is euchromatin, black is heterochromatin. My question was embryonic stem cell versus differentiated cell, which one is whiter?
Embryonic.
It should be embryonic stem cell, right? Embryonic stem cell may be white angel, and then differentiated cell may be the black pebble-like cells. Okay? You can, kind of exaggerating this picture, then you can draw it that way. So if you look at this structure now by looking at those structure, this heterochromatin, it may not be only one gene, right? So total gene number in this chromatin is maybe 20,000 or 30,000. There one chunk of gene here. Maybe this is easily hundreds or 200s of a gene cluster. And if they are close together physically, then they may be related to affect each other, and these genes maybe is kind of the same functional groups we can expect, right? That's why if you add this kind of a HDAC inhibitor is open up this structure, then HDAC inhibitors go in. It may not open up every side. Maybe it can open up maybe this area or this area. If this area is unique in cancers, then this area genes maybe most likely they can be tumor expressed genes, and they are suppressed by these epigenetic changes. Okay, if you add epigenetic modifier like the HDAC inhibitor, usually this is kind of a biochemical pathways we have studied over last 100 years. Looks quite complicated, and each small spots there are enzymes. And then maybe this line or cycle, this is kind of some biochemical processes we discovered so far. Looks like complicated, but how many enzymes are here? Still maybe 1,000 or 2,000. This whole information is less than 5% of every gene we have in our body. Okay. So far, the drug discovery has been used. Okay, this gene is a problem. That gene is mutated. That we need to make inhibitor or activator for the gene. That is kind of conventional drug discovery or drug targets. Okay, but if you use epigenetic modifier, as you have seen in the previous pictures, it may not affect only one gene. It affects big chunk, like okay, this area is all activated or all suppressed. So this is kind of a chunk by chunk. That effect is huge. We didn't cover that much for this week, how we make IPS from differentiated cells. Okay, simply speaking, by adding this kind of changing heterochromatin structure into euchromatin structure, by adding this epigenetic modifier. Okay, young cells is wider. Older cells, they become darker and darker. To change this older cell into the young cells, by adding this kind of sometimes protein factors like Yamanaka factors or some chemicals which can change this heterochromatin into euchromatin. And their change is not only one by one, they change hundreds to thousands at the same time. That's how we make epigenetic changes and also generating stem cell from non-stem cells. Okay. So this epigenetics is a relatively new field, relatively new, maybe about 10 years of history, the real drug development. But the impact and also those approach is huge and also very different approach from conventional approach. So that's why this is quite important topic. And because of those limited the time, I cannot teach you everything about this. Maybe you can do 2-3 the weeks of lecture for this. But I leave this to you by reading that epigenetic book. Okay. Long question. Okay, there's some point here, but some points is not fully the correct point. You try to digest the question first. If there's a short man and short woman, and then the baby, well, if the baby is a high, then by changing those methylated pattern. But epigenetics, environment, and individual efforts can switch on those genes. It's difficult to believe. Okay, from father and mother, we get those genes, and father is the child, mother is the child, and the baby, you may say by changing those methylation pattern and change it, but is that really possible to overcome those heredity? Well, very interestingly, let's just say if you look at the pictures, one example can be maybe Vietnam. When I was young, I still remember I have seen in the newspaper, Saigon was conquered by the northern the Vietnamese. That was in 1975. The moment I was 7 years old, but I don't know how I can remember that. I have seen it in newspaper. And at the moment, always when we see those Vietnam people, they are kind of shock people. 30 years later, when I went to the US, when I met Vietnamese, the young guys, maybe second generation in US, I never seen short people. Most of them they are taller than me. Similar the feeling when I got back to Korea after 20 years of outside the living, and new generation of young people, they are much taller than before. Those environmental changes, not only nutrition, but those environmental change, environmental impact, those the outcome phenotype changes, that's huge. Exactly how, that's difficult to say, but within one generation, within 30 years, okay, that's a too short time to change those the genetic the information. But not only nutrient effect, somehow those gene regulation effect is huge. So that's why epigenetics is kind of a new important field. There are many things we do not understand. One thing also important fact is, when I was young, my grandmother told me that she was not very well educated. She says, baby's bone and muscle is inherited by father, and skin and eyes are inherited by mother. I didn't tell her, but I thought, hey, you did not get proper education, so it doesn't make any sense, I thought. But more and more, this epigenetic modifying, the modification and also effect, is significantly difference. Even though the man and human, except X and Y chromosome, we believe all of those genes are same, but it's not. Many genes from father, and they are all activated. Some genes from mother, they are only from mother side the gene is activated. So there is a significant impact of whether this gene is that came from mother side or father side. It's not equal one-to-one the competition. They are very significant the effect. Some of our characteristics is from mother side, not only mitochondrial DNA, as we mentioned last time. Okay? even our the genomic, the impact. There are many things we didn't know before. That's why, the book will give you some new possibility and new way of thinking. So I'm telling this 3 times today. It's a very good book to read. Okay, next question. If DNA is damaged, whether that damage can inherit to next cell or next generation. If that damage is somehow cured, and then that damage most likely, sometimes it can change the sequence, then mutation can happen, right? If a mutation can happen and if it's becoming one of those ATGG, then yes, that mutated sequence will be inherited. If that damage is too big, then it cannot be fixed, then it will be cleaved. Then those cleaved sequences can go on, or maybe that cleavage is critical, then the cells will die. Okay, so small damages we try to recover, even though the sequence is slightly changed, then mutation happens and that information can transfer. If that damage is too high, then the cell cannot survive. I'm afraid our time is almost over. There's several more points, but we cannot cover everything for today. Okay, let's stop here. This lecture. Anyway, this is the last the lecture before the midterm exam. So exam will be next week, May 7th, Thursday. So we will meet here at Zoom class, and I will give you the guide how to take the exam. As of today, it's better for you to fully perform yourself. Be prepared with the good machines. Maybe just only the cell phone may be difficult. At least the computer, the notebook, and be ready to come to the class. And I will give you the guide. And during those until those the meeting exam, I will open the question session on the website. So try to put up the questions there, or if it's urgent, then you can give me the questions to cut on. Okay. Any question for midterm exam?
Actually, when is the end? When does the test end?
Let's keep it time as our class time, not to collide with your exam schedule. So we start at 2 p.m. then maybe 3:15.
Thank you.
Okay, if no more further questions, then okay, have a good day, guys, and enjoy the midterm exam. Exam's purpose is not torturing you. This is to give you a chance to summarize everything you have learned and reorganize and then systemize it in your the brain to keep it along. Okay, guys, thank you and see you next week.